Let's look into OLC, or Overlap Layout Consensus method. We'll talk about different steps in it. We'll look into some advantages and some limitations, and we will also see there are some OLC based assemblers. OLC is the most successful assembly so far. That's what they claim. There are 3 steps in it. Number 1 is overlap stage, and number 2 is layout, and then the third one is developing a consensus. So in overlap, we take the reads, we compare them with one another, and we try to find their overlaps. And then using this information, the overlap information, we construct the overlap graph. So here is the overlap stage. We take read 1, read 2, and both of them, they overlap in their terminal regions, the ending part of first one and beginning part of the second one. That's what we want. During this stage, layout stage, overlap graph is analyzed in order to identify the paths that correspond to the genome being assembled. So now we have overlaps, and we place those overlaps in the shape of a graph. So within the graph, we are trying to find those paths. The goal is obviously to find the single shortest Hamiltonian path, so that visits each node in the graph exactly once. And in the consensus stage, what we do is, so in the end, we can get those reads, we can get those assembled genomes, and we can get those overlapping reads and put them together and develop their consensus. So, and then we try to compare that consensus with our assembly. So that is the consensus stage. This is what is happening in this technique. So we take those reads, we give it to our algorithm, we identify some overlaps, and we identify those regions which are overlapping. Then what we do next is we identify the reads which are connected by different overlaps. So in this way, we take those reads. So for example, this read can be connected to this one, can also be connected to this one. So in this way, we put those reads and we get all their connections like this. So in the end, we get many paths, many connections. We try to simplify them and identify a Hamiltonian path, as we do here in this diagram. And in the end, we develop a consensus sequence. So this is what it's happening in overlap layout consensus. Advantages. So it is suitable for large set genomes, works for both short and long reads, and is capable of handling the NGS data. That's what we are talking it about. It generates correct order of context and recognizes repeats, but the issue is it collapses them together. So sometimes the repeats are there, especially in case of the tandem repeats. So they are put together and they are, collapsed into just one repeat. This is what we see in this example. So these all B's, they go together as one B in the final assembly. Limitations, the processing cost. Obviously, in order to do these overlaps, you need to compare each read with every other read. So this is it takes too much computation. So, and same way, it takes too much time to do all those pairs of comparisons. And another limitation is, in order to find the Hamiltonian path, different algorithms which are used, they are not that efficient. So there is another issue with that. Different assemblers which are using this approach: Arachne, you might have heard of it in the literature. Then there is Celera assembler, we talked about that. Nubler, we also mentioned in a previous slides. Edina and CAP and PCAP. So OLC is the most successful assembly so far, though not the most, optimal or the finest one, but amongst what so far we have seen, amongst the other assemblies, it's being propagated as the better one.